Our current topic is regulation of eukaryotic gene expression. And we've talked about regulating access to the DNA sequence by epigenetic regulation. And then we talked about, okay, once you have this accessible naked DNA, the thing that regulates whether or not transcription occurs are activating transcription factors or silencers, but mostly it's activating transcription factors that bind to enhancer regions and promote transcription of nearby genes. So that was transcriptional regulation. So sort of pre-transcriptional regulation, transcriptional regulation, but we can also regulate the RNA itself. So regulation at the level of the RNA, we're going to refer to as post-transcriptional because transcription has already occurred. So post-transcriptional regulation of gene expression. So what can happen at the level of RNA that can change going from a gene, the genetic DNA sequence, to the type of functional protein that's made? Well, we already have hit on some of these. One of those is that we can have that alternative splicing. So alternative splicing where you have this differential removal of introns, and it results in different flavors of proteins. They're proteins that are all derived from the same gene, but they have different functional aspects of them based on whether, which exons stay in the mature message to be translated into protein. So we've already talked about alternative splicing, but just to kind of reiterate that it is a way that we regulate what type of protein outcome we have from transcription of the gene. Another thing is that not all messenger RNAs are the same. They have half-lives. So an mRNA half-life. This varies based on the mRNA, so different mRNAs have different half-lives. What determines the length of the half-life isn't 100% completely known, but it has to do with the length of the poly A tail, different sequences that are in the message itself, and other sort of modulatory molecules that we're talking about, or that we will talk about. So different mRNAs have different half-lives. And if we look at this chart down here, it shows sort of, the common half-lives for different RNAs. And look, it's on the order of, a couple of days to a few days. So it's really measured in terms of hours. The shortest lived have half-lives of, less than 6 hours, 6 to 12 hours, et cetera. So, we can really think about this in the range of hours to days, depending on the messenger RNA. So the half-life, just like you've learned about with radioactive decay and stuff, half-life is the time it takes for 1/2 of the message to degrade. Now, what does that mean to protein expression? Well, if we have a message here, as long as it's around, ribosomes are going to be popping on this thing, continuously translating it. So shorter half-lives ultimately results in decrease or lower protein expression, because the message is not around as long to continue to be translated to make message. Now, another thing that can influence the half-life and the degradation of messenger RNAs are small molecules that can interfere with the message. And we call this process RNA interference. So this is where small RNA molecules called siRNA in prokaryotes and used in the lab. Si stands for small interfering. RNAs, or in eukaryotic cells, our endogenous natural sort of interfering RNAs are called miRNA, which stands for micro. So they both ultimately end up doing the same thing. Their initial size and or where they originate from differ a little bit, but we're going to use them interchangeably. So what they do is small interfering SI or microRNAs bind to, or they promote, they block translation and or promote degradation of the message. So I'm going to kind of give an overview of how this process works, regardless if it's SI or micro. Like I said, they have some differences, but we can think about them at the same thing. So the interfering RNA has to be transcribed from the genome. So there are coding regions within the genome, especially in eukaryotic cells. There would be swaths of the genome, areas of the genome that have information that code for these functional microRNAs. So the RNA has to be transcribed. In bacterial systems, it would be siRNA. In the lab, we would purchase the RNA from the RNA store. And depending on the type, it has to be cut by a special enzyme. The RNA is cut to a specific size by an enzyme called dicer. Kind of a fun, like dicing up the RNA, cutting it. So here's our RNA being pre-mRNA or pre-siRNA, pre-microRNA, sorry, or pre-siRNA being transcribed from the genome. It's transcribed as a double strand. Let me put that in here. Double-stranded RNA is transcribed from the genome. It gets cut by dicer. It binds to a protein complex, forming what's called the RISC complex. RISC. One of the RNA strands degrades. Let's look over here. Here's our RISC complex. This is now a single-stranded RNA. So I'm going to say it's leaving a single-stranded RNA guide strand. Well, what is it guiding to? Well, this is it can bind to anything that it's complementary to, and it binds to the RISC with the guide strand. To messenger RNAs that are complementary to the guide. Notice it's not the entire messenger RNA, it's just a portion of it. And now this RISC complex can chop up the RNA, or it can block the ribosome from proceeding for translation. And I have an animation.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future, perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of the cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and they're important in a multitude of biological processes and as tools continues to be revealed.
And just to reiterate that these RNAs are coded for in genomes, depending on what type of genome we're talking about. So just as an example, in eukaryotic cells, we have endogenous microRNAs, meaning microRNAs that are actually coded for in our genome. So some parts of the DNA code for proteins, and they are transcribed into messenger RNA. But there is also information to make these microRNA molecules that can go and then join with the RISC complex and target messenger RNAs for degradation, shortening the life of the messenger RNA. So genes that have to respond really quickly to environmental conditions, so a lot of sort of inflammatory gene products have microRNAs that target those messages so that they can be downregulated really quickly. So it's really kind of this cool phenomenon. So now we've looked at epigenetic regulation, transcriptional regulation, post-transcriptional regulation. At the level of the protein itself, we can also have regulation, regulating whether or not the protein is active, regulating whether or not the protein, how it works. I'm not gonna lie, this is my favorite type of regulation. It plays a big role in signaling, and it is post-translational regulation. So after post-translational, after translation, post-translational regulation. We can also call it post-translational modifications of proteins. So how can proteins be modified to change what they're doing inside of a cell? One thing is they can have different organic groups attached to them. And we call these covalent modifications. So many proteins have to be modified by the addition of or removal of additional functional groups or other types of small molecules. So many proteins have their function altered by the covalent attachment of some small molecule, functional group, or some other molecule. Some other non-protein molecule. So what are some types of post-translational modifications? Some types of covalent modifications. So I said this is kind of, post-translational modification is my favorite sort of class of regulation. This guy here, the phosphate, is my favorite type. So phosphorylation of proteins is a really common way that proteins can be modified. We actually have already seen this once with histones, right? We can have phosphorylation of histone groups which help to open up the chromatin in epigenetic regulation. But a lot of enzymes are also modulated by the addition of a phosphate group. So phosphorylation is the addition of a phosphate group. And often they can act, so it often acts as an on-off switch for protein activity, specifically enzymes. So a lot of our signaling molecules are modified by phosphorylation. So what do I mean by that? Let's take a look here. So here we have a protein, generic protein, a phosphate group, so specific residues can be phosphorylated. So on proteins that are modified by phosphorylation, specific residues, either tyrosines, serines, or threonines. So these are the 3 amino acids that have the potential to be phosphorylated. So on protein specific tyrosine, serine, or threonine residues can potentially be, or potentially have a phosphate added. That's the process of phosphorylation. The enzymes that add phosphates are kinases. So kinases are enzymes that take a phosphate off of ATP and they plop it onto a specific location on a protein. So this can either potentially turn on or off enzyme activity, depending on what the protein is. The phosphates are removed. The process is called dephosphorylation by enzymes called phosphatases. So there are a variety of different kinases and phosphatases floating around in the cytosol in our cells. And when they get turned on, they can quickly add a phosphate group. Kinases can quickly add a phosphate, phosphatases can quickly take it off. So let's look at a specific example. Like I said, it depends on what the protein is, whether or not phosphorylation turns it on or turns it off. Here's an example of a specific protein called glycogen phosphorylase. So this is involved in carbohydrate metabolism. So we have glycogen stored in our muscle cells. So if we need to have glycogen, the glucose is liberated from glycogen, we need glycogen phosphorylase. Well, this happens to be an enzyme whose activity is controlled by covalent modification with phosphorylases. So let's take a look at the structure. Look at the helices. Remember our protein structure that we touched upon? It's loaded with helices. This is a homodimer. So we've got one subunit, two subunit, but they're the same. And look at here, we've got a residue, serine 14. Serine 14, because it's a homodimer, so we have two of those. And here's a little hydroxyl group on serine. In an unphosphorylated, dephosphorylated form, glycogen phosphorylase activity is off. It is not liberating glucose from glycogen. It is off. And look at where this serine is. It almost acts like an arm. It's blocking access to the active site. So the active site is in here. That's where the glycogen is going to go in. The glycosidic linkage will be cut. But when it's not phosphorylated, it can't get in there. So the attachment of a phosphate group, which you know is like a little negative, it's like a little chunk of negative chemical that's popped on there. What it does, it swings out that arm and it opens up the active site. And so phosphorylation at serine 14 activates glycogen phosphorylase. So that's a specific example of how phosphorylation of a particular enzyme can control its activity. In this case, the phosphate is acting as an on switch. Removal of the phosphate by phosphatases would turn off the enzyme. So phosphorylation isn't the only type, even though it's my favorite type of covalent modification. Some proteins we can have the addition of, we've talked about acetylation. So addition of acetyl groups, of methyl groups, and other functional groups, right? So these are all types of functional group modification, but we can also have the attachment of other organic molecules. We can have the addition of carbohydrates, or sugars to proteins. This is really common for surface proteins. So proteins that are embedded in the membrane of cells very often have carbohydrate covalent modifications. If you remember the ABO blood type that we talked about before, that is carbohydrates on the surface of a protein on red blood cells. So what do we call proteins that are modified by the addition of carbohydrates or sugars? We call those glycoproteins. Some proteins are modified by the addition of a lipid group. Now, what is a chemical characteristic of a lipid? Here's a kind of a generic cartoon depiction. Well, these tails are very hydrophobic. So imagine you've got this soluble protein floating around in the cytosol of the cell, but you can, if it's attached to a lipid, that lipid can anchor it to a plasma membrane, either the plasma membrane or the membrane of organelles. So the addition of a lipid adds a sort of hydrophobic tail to proteins and can change its localization within inside the cell. We call proteins that have lipids attached to them lipoproteins. And there's other types of covalent modifications too, but these are some of the common ones. Some proteins, when they are synthesized, when they are translated in an inactive form, and they need to be cut before they're active. So we'll call that proteolytic cleavage is necessary for some proteins to become active. So some proteins must be cut. Or we can even processed is another sort of term that we use for that. So some proteins must be cut or processed to become active or to become functional, to do their normal function. So here's an example here. Well, I will say in that case, if a protein is translated in this inactive larger form, we call that a proprotein. That undergoes cleavage to becoming the active form or the functional form. So an example of that is insulin. So here's the sequence, the amino acid sequence of insulin. We've got the C terminus and the N terminus of insulin. Before, this is not the functional thing that goes into your bloodstream that allows the cells to take up glucose. For insulin to become functional, this pink portion has to be cut and removed. And here is the functional insulin. This is the thing that acts as a hormone in our blood and signals, binds to insulin receptors and tells those cells to pick up glucose. And then we can also have protein degradation, just like we had for sorry, I'm having some technical issues. There, I think I resolved my technical issues. So where was I at? So protein degradation is another way we control sort of gene expression, having something functional going on. And because proteins also have half-lives, they don't stay around forever. So proteins have half-lives just like messenger RNA does. Now they tend to last a little bit longer. They tend to be more stable than messenger RNAs. So they tend to have a half-life in the range of days to weeks. But ultimately, all proteins are going to get broken down. They're chemicals. Those bonds, those peptide bonds are going to get broken. We call the process of protein degradation is called proteolysis. And so this is where proteins are degraded or chopped up into smaller peptides and ultimately back to the amino acids that made them. And then these amino acids can go on and be recycled to make more proteins. So just here's some common half-lives. It looks like, you know, in the range of 5 to 7 days, but some proteins, some types of antibodies have a half-life of 3 weeks. And so if the message has been degraded and you're not translating any more protein, right, that protein itself sticks around and does its job. But ultimately over time, if you're not translating more of it, if it's not continuously expressed over time, the levels of those proteins will decrease. And that's what you hear with the COVID vaccination and you measuring antibodies in the blood will act over time. The antibodies are going to decrease because they're just proteins. So if they're not being actively made, the protein levels, the antibody levels will decrease in the blood. That doesn't always mean that the immune system has lost the ability to respond quickly. You can still have memory cells that can be activated and jump into action really, really quickly when they're challenged with the infection again. But I digress. So the point is this, that gene regulation in eukaryotes is complex. We have to have the ability to modulate whether or not we're getting those proteins, those molecular machines or not. And so they're regulated at all sorts of levels, epigenetically, transcriptionally, post-transcriptionally, and then at the protein level itself, post-translationally.